Hi, my name is Alexsis Wilson, and for my local Expert 3 project, I'm going to be talking to you about stem cell culture. And this is all going to be in relation to our Module 5 of our Introduction to Cell Cultures, which we talked about in the lecture. Obviously, I'm specifically going to talk about stem cell cultures here in this video. So stem cell culture is really as it sounds. It's the growing of stem cells in a lab. Stem cells can proliferate in laboratory environments in a culture dish that contains a nutrient broth known as culture medium. And most stem cells attach, divide, and spread over the surface of the dish. The culture dish becomes crowded as the cells divide, so then they are going to need to be replated in the process of subculturing, which is repeated periodically many times over many months. And each of the cycles of subculturing is referred to as a passage. And then these cells can be transformed into a variety of terminally differentiated adult cell types, which can then be used for disease modeling or toxicity testing. So then there are two types of stem cells that can be cultured. You have the pluripotent stem cells, which are then further divided into embryonic and then induced pluripotent stem cells, or iPSCs. These stem cells are those that have the ability to differentiate into any type of cell in the adult body. And then you have somatic or adult stem cells. These are found in a tissue or an organ and then can only differentiate into the specialized cell types of that tissue or organ. Different types of stem cells often require different culture conditions. For example, human-induced pluripotent stem cells and human embryonic stem cells must be cultured on coated plates with a supporting layer of feeder cells such as mouse embryonic fibroblasts or human foreskin fibroblasts, or on an extracellular matrix such as a basement membrane gel. Other examples of stem cells include neural stem cells, which can differentiate into various types of neural cells, and hematopoietic stem cells, which can give rise to different blood cell types. So when you're choosing what cells you want your stem cells to then differentiate into, it's very important to know that going in so that you can start with the correct type of stem cell that you need. So then going right into some works that have been published that are using stem cell cultures. The first figure here having stem cell cultures analyzed was in a paper titled Exploring the Regulation of Human Neural Precursor Cell Differentiation Using Arrays of Signaling Microenvironments. So here, Owen and the coworkers generated libraries of the ECM components and growth factors to screen their computational effects on the stem cell differentiations. Using a piezoelectric arrayer, the pre-mixed combinations of signaling molecules, including growth factors, ECM components, cell adhesion molecules, and the morphogens, were printed onto the arrays. The primary biopotent human neural precursor cells were then subsequently cultured onto those arrays. And then the proliferation and differentiation of the neural progenitor cells was examined with immunostaining, followed by quantitative image analysis. And basically what they found was that the co-stimulation by WNT and Notch was shown to maintain the cells in an undifferentiated state, whereas bone morphogenetic protein 4 induced an intermediate differentiation phenotype characterized by simultaneous expression of the glial and neuronal markers. The next paper here wanted to investigate the effects of high cyclic hydrostatic pressure on cellular viability. To do this, single-cell suspensions were exposed to different levels of HHP ranging between 5 and 250 MPa for a short-term period of 10 minutes. After 24-hour morphological analysis provided that cells which were under the HHP between 5 and 100 MPa were fully attached and showed a normal morphology after pressurization, indicating that the selected pressure levels did not impact the cellular adhesion properties. There was also no significant difference in metabolic activity of the cells which were exposed to the HHP between 5 and 100 MPa, which confirmed the morphological observation. Moreover, the quantitative evaluation of the cell's viability immediately after pressurization process revealed that cells subjected to HHP of 5 to 100 MPa showed a high rate of metabolic activity, which can be seen in figure 1c. However, despite showing high metabolic activity in a 100 MPa group, morphol- morphological analysis after 24 hours revealed the presence of some clusters of cells which exhibited a rounded morphology. On the other hand, analyzing both tests demonstrated that the HHP at the magnitude of 150, 200, and 250 MPa was extremely lethal to the cells, where a significant difference in metabolic activity and a round shape of morphology were observed. In line with those results, it was reported that the cells exposed to the pressure above 200 MPa showed a round-shaped morphology, and it did not adhere to the culture dish even after 24 hours. The final figure that I want to show here is from a paper having to do with multipotent mesenchymal stem cells from adult human synovial membranes. And here they wanted to look at the in vitro expandability with limited senescence of the synovial membrane derived cells. So SM-derived cells cultures were initiated as primary cultures of cells that grew out of SM-derived cell suspensions by selectively attaching to tissue culture plastic. After the first passage and throughout in vitro expansion, SM-derived cells appeared microscopically to be a relatively homogeneous population of fibroblast-like cells. The stem cells displayed a long-term self-renewal capacity, so then they investigated the in vitro expandability of the SM-derived cells from donors of various ages. Starting from the first passage, they analyzed the kinetic growth of SM-derived cells from all donors tested. Figure 1a and b shows that although no significant telomerase activity was detected, the growth curve of SM-derived cells from 2 donors was linear up to 30 population doublings. In all donors tested, the SM-derived cells were expanded over at least 10 passages with a similar kinetic growth. They then wanted to examine the cell senescence. They analyzed SA beta gal activity in SM-derived cells from all donors during expansion. What they found was that at early passages, less than 1% of the cells displayed detectable SA beta gal activity. Weak to moderate B gal staining of a few scattered cells was then observed at late passages. Finally, I want to talk about some advantages and disadvantages to using stem cell cultures over primary cells in research, since we just looked at some of the research dealing with the stem cells. So, some advantages to using stem cell cultures over primary are that stem cells have the unique ability to self-renew and differentiate into specialized cells. They also offer an unlimited supply of cells that can be used for research and drug development, and they can be used to generate specific cell types that are difficult to obtain from primary cells. And then also the iPSCs can be derived from donors with known disease phenotypes, whose cells can then be used in a patient-specific disease model and drug screens. Some disadvantages to using stem cell cultures over primary cell cultures are that they are more costly than the primary cells. If not isolated and differentiated properly, the risk of forming tumors can also occur with stem cells. Primary cells are also more representative of what is occurring in vivo and can be used to model complex physiological behavior. Thus, differentiation and maturation protocols require improvement before stem cells can be considered accurate models of human physiology. However, stem cells can be expensive to obtain and maintain, and there are ethical concerns surrounding their use. Human embryonic stem cells must be derived from human embryos, and destroying these embryos makes their use ethically controversial, which in turn has also led to an increased regulation of their use in research. There is a lot, of course, that scientists and researchers can still learn about stem cell cultures and how to utilize them in drug development and discovery, but the possibilities of where they could take them and what keeps the drive going into learning more about them. The prospects are exciting, and the future of stem cells is something that should be watched. And then finally, my references are listed here for my presentation. Thanks for listening, and I hope that you found some of this interesting and informational. Bye.